So last week we talked about the ways in which the electrical impulse gets propagated down the axon, and what about the neuron allows it to even generate an electrical impulse. For this week we're going to be talking about communication between neurons, and that's called synaptic transmission. So we'll be delving into the aspects of the neuron that allow it to communicate with other neurons, and where the different ways in which the chemical messengers allow for variation in response from the receiving neuron. So let's get started. You know that the information is carried in the neuron in the form of electricity, what we've been calling the electrical impulse, but is also known as the action potential. The information, unfortunately, for it to be useful in any way, needs to be passed on to other neurons. So one of the ways it can happen is through the direct exchange of ions in what is typically found in electrical synapses. And we're not going to be spending much time on electrical synapses because the vast majority of synapses that are found in the vertebrate nervous system are those of the chemical kind. So if you're curious about the electrical impulses or electrical synapses, which involve direct exchange of current from one neuron to another, allowing for synchronized behavior between neurons, I'm happy to talk to you about it. But the vast majority of this course is going to focus on chemical synapses. So we're going to be covering this in greater detail than electrical synapses. But I wanted you to be aware of the fact that electrical synapses existed. So neurons, right, once the action potential reaches the axon terminal, in some way this message that's in electrical form needs to be communicated to the receiving neuron. The form in which the information passes actually changes. So it passes through the neuron in electrical form. However, the way in which it's communicated to the receiving neuron is in chemical form. And then once it enters the receiving neuron, it takes on electrical form. And again, we'll be talking about that transition or transference of information. But you should know that within the neuron, the information being carried is in the form of electricity. However, between neurons, this information is modified to take on a chemical form. So the chemical synapse has unique properties to it, and we're going to be discussing it here. So the chemical synapse consists of the membrane of the presynaptic cell, which is what we call a cell sending the information, the postsynaptic cell, which is the cell receiving the information, as well as the space between them, which is known as the synaptic cleft. So technically, a synapse isn't just the space between the two neurons, rather it involves characteristics of the presynaptic membrane, the synaptic cleft, as well as the postsynaptic membrane. So these three components are what make the synapse. You might find in the terminal button or the axon terminal a ton of mitochondria. If you remember from several lectures ago, the mitochondria serves as the energy source for everything that happens in the cell. And so this tells us that the process of making or releasing chemical messengers is a very energy draining process if you need mitochondria so close to where all the action's happening. You'll find very small circles, what look like circles, containing chemical messengers or neurotransmitters. So there are two types of vesicles, which is what we call these packets, the containing neurotransmitters. There's small and larger containing neurotransmitters. The smaller containing neurotransmitter vesicles, so vesicles are the pockets of neurotransmitters. They store neurotransmitters until they're given the signal to release. The difference between the small neurotransmitters and large neurotransmitters is that these larger vesicles contain chemical transmitters that are actually proteins. They're called neuropeptides. So proteins are larger in structure, they take up more space, which is why their vesicles tend to be larger in size compared to what we think of as classic neurotransmitters like serotonin or dopamine that you might have heard of. So these are synaptic vesicles. And the point at which these neurotransmitters are released is known as the release or active zone. They happen to be right over what is known as postsynaptic density, which in fact are proteins that detect the message contained within the neurotransmitters. So these neurotransmitters are communicating a message to the postsynaptic cell, and the way in which they do that is by binding to proteins that serve as receptors. So they work kind of like a lock and key mechanism, and so the receptors are all collected together right underneath the active zone so that when the neurotransmitters are released, they don't have to travel very far to bind to and activate the receptors on the postsynaptic cell to send the message along. So here's another image showing exactly the same thing. We've got tons of mitochondria found within the synaptic terminal because this is a very energy hogging process. We've got synaptic vesicles that are waiting and ready to be released right over the receptors, these proteins that are responsible for detecting the neurotransmitters released from the presynaptic membrane. So what I've shown you so far were just diagrams of what the synapses look like. What you're looking at here are real synapses. So these are images collected from a very powerful microscope called an electron micrograph. And what they use, the technique is not so important, but it allows us to see very small, incredibly difficult to see under a regular microscope images. And so what you're looking at here is an axon terminal in the central nervous system and an axon terminal in the peripheral nervous system. So what you're seeing are these dense core vesicles in the peripheral nervous system. They are the ones that contain the neuropeptides, the neurotransmitters that are actually proteins, whereas the vast majority of the neurotransmitters in the central nervous system tend to be of the classic neurotransmitter nature. But all of these little circles that you're seeing around here and on the bottom here are synaptic vesicles. They are filling the terminal. Additionally, you see tons of mitochondria in both the central nervous system and the peripheral nervous system, again supporting this idea that releasing neurotransmitters uses a ton of energy. So this little area here, you're seeing some neurotransmitters being released because they look like black substances. There's so much of it. And right underneath them, you can kind of sort of see the synaptic cleft, but not really, because so many neurotransmitter molecules are being released. But right underneath them, you see a collection, right? That postsynaptic density I was talking about earlier, those are all receptors for the neurotransmitters that are being released. So I absolutely love this image because this is an actual picture of a synapse, and you can see how many synaptic vesicles and mitochondria are actually located in a synaptic terminal. There are many, many different types of synapses that you can find between neurons. Common synapses are those demonstrated here on the right. Axodendritic are between a terminal and a dendrite or dendritic spine. So the presynaptic terminal releases neurotransmitters onto a dendrite or dendritic spine. You can have an axosomatic synapse where the presynaptic terminal will release neurotransmitters onto the cell body directly as opposed to just the dendrite. And then lastly, we can have what's called an axoaxonic synapse. So this is where an axon terminal releases neurotransmitters directly onto either the axon or another axon terminal, effectively controlling the action potential or the release of neurotransmitters. And we'll revisit axoaxonic synapses later on in this lecture. But these are typically the kinds of synapses that occur between neurons: axodendritic, axon and dendrite, axosomatic, between axon terminal and cell body, and then axoaxonic, between axon terminal and another axon terminal, or axon terminal and an axon directly. So when the message gets transferred into chemical, it, as I mentioned, once it enters the postsynaptic cell, it gets reverted back to electrical again. So I don't know if you remember from a while ago, but when Hodgkin and Huxley stimulated the giant axon of the squid, when they fed a current that was less than the threshold of excitation, they found a slight increase in the membrane potential. Well, we're going to be talking about those slight increases in membrane potential right now. When it happens at the level of the synapse, in the postsynaptic cell that's responding to the presynaptic cell's neurotransmitters, it's known as postsynaptic potentials. So neurotransmitters are released from the presynaptic membrane, they bind to the receptors on the postsynaptic cell, and then depending on the type of neurotransmitter and its receiver or receptor, again, remember the receptor is a protein, one of two things can happen. You can either have a depolarization, which is the membrane becoming more positive, the membrane potential becoming more positive, and this is known as excitatory postsynaptic potential. So again, hopefully you'll remember this image from the previous lecture, but these increases in membrane potential, if they were happening at the level of the synapse, would be considered excitatory postsynaptic potentials. They are depolarizations, but they are considered excitatory postsynaptic potentials. The other thing that can happen is known as hyperpolarization. So when the membrane dips below the resting membrane potential and becomes more negative, that's known as hyperpolarization. So this might be confusing because you might think of hyper as going up, but in fact when you think of polarization, it means two things are going further apart from each other. So when something's getting hyperpolarized, it's getting further away from, in this case, the threshold of excitation. So hyperpolarizations, when they happen at the synapse, are known as inhibitory postsynaptic potentials. They're called inhibitory postsynaptic potentials because they drive the membrane potential further away from the threshold of excitation and further more negative compared to the resting membrane potential. So again, here are just some examples. EPSPs, or excitatory postsynaptic potentials, increase the likelihood of an action potential happening. So if you think of the membrane, right, needing to reach that threshold of excitation, if you have an EPSP, it's going to get closer to that threshold of excitation value if there's sufficient EPSPs, if there's sufficient increase in the membrane potential. IPSPs, on the other hand, drive the membrane potential away from the threshold of excitation, so they decrease the likelihood of an action potential occurring. So here we go, right? Here's a hyperpolarization. If this is the threshold of excitation, it's causing the membrane potential to move further away and become more polarized away from the threshold of excitation. What's different about postsynaptic potentials and action potentials is that postsynaptic potentials can demonstrate graded responses. What I mean by graded responses is that they can be little or big. Action potentials, if you recall, do not change in size or intensity. That's not the case for postsynaptic potentials. They can actually change in size and intensity. They can be graded. They typically start at the level of the synapse and then travel passively as charges diffuse into the postsynaptic cell. So whenever we talk about EPSPs or IPSPs, we're talking about the membrane becoming more positive or more negative. But just like the membrane potential or action potential, right, that changes in charge, these are the result of ions moving into or out of the cell. So when these ions enter the cell, they start at the synapse and they diffuse passively and go towards where there's less of them. Okay? So postsynaptic potentials are not regenerated like action potentials through the opening of any channels. They start at the synapse and then they just diffuse through the neuron. What is important to note is that no single postsynaptic potential has any effect on the action potential or the likelihood of the action potential occurring. You actually need collections of postsynaptic potentials in order to reach the threshold of excitation. So when a neuron fires, or decides to fire or have an action potential, it's the result of net combinations of the net effect of postsynaptic potentials reaching the threshold of excitation. And this decision to fire or to have an action potential starts at the axon hillock. This, arrows pointing to the kind of head or tip of the axon, and the decision to fire happens at the site. The reason why it happens at the site is because it's the first location where you're going to find those voltage-gated sodium channels, and they're the ones that initiate action potentials. So just keep in mind, in order for a neuron to decide to have an action potential, the membrane potential needs to reach the threshold of excitation, and the threshold of excitation is only going to be reached if the sum of all of the postsynaptic potentials this neuron receives reaches that particular value. So let's talk about how that happens. So when you add together all of the graded EPSPs and IPSPs at the axon hillock, the decision to fire or not occurs. Combining the signals, combining all these EPSPs and IPSPs into one overall signal is known as integration. So if a neuron receives a lot more EPSPs, it's going to get more positive. If a neuron receives a lot more IPSPs, its membrane potential is going to be more negative. So overall, if a neuron has more EPSPs compared to IPSPs, and this combination of EPSPs and IPSPs reaches the threshold of excitation, an action potential will likely occur. However, if a neuron receives more EPSPs, so more hyperpolarization, more negative values than excitatory values, it's quite unlikely that the threshold of excitation is going to be reached, and therefore it's unlikely to have an action potential. So this is kind of a visual of how this might happen. If a neuron, right, it's got 4 contacts, 1, 2, 3, and 4. If, let's say, these 2 synapses or contacts it's making are excitatory, and this one here is inhibitory. So if these 2 synapses are activated, it's going to generate tons of EPSPs, and at the axon hillock, it will decide to have an action potential or fire because the threshold of excitation will be met. On the other hand, if you have a combination, equal amounts of IPSPs to EPSPs, by the time it reaches these passively diffusing ions reach the axon hillock, the IPSPs may have canceled out the EPSPs, resulting in no change in the membrane potential, and therefore no action potential will be generated. So the idea of kind of combining like charges is known as summation. Where's my little thing? Is known as summation. When you kind of combine all the positive charges into one greater positive charge, that's known as summation. You can also have IPSPs summating. So you can have an overall combination of IPSPs, and that will be considered IPSP summation. It happens exactly the same way. It's just easier to see it with an EPSP. So a single synapse, right, a single axon generates an action potential and activates this synapse. You'll see a slight increase in the membrane potential to a more positive value. Not surprising. If you have 3 different synapses activated at the same time, releasing neurotransmitters and generating EPSPs on this particular dendrite at the same time, you're of course not surprisingly going to see a much larger EPSPs. When you have 3 different or 4 different or 2 different synapses activated at the same time to produce a larger EPSP, that is known as spatial summation. You can also have what's known as temporal summation, where a single synapse gets activated repeatedly, back to back to back, and then the effect on the postsynaptic cell is that you end up seeing it piggybacking onto each other, but ultimately you have an EPSP large enough to produce, to reach the threshold of excitation. So both of these, spatial summation and temporal summation, represent a larger EPSP being generated as a result of multiple EPSPs combining into one. It can either happen over the course of multiple different synapses, or you can have one synapse activated repeatedly to produce a larger EPSP. So your textbook kind of represents all of this in this one particular image. So what it's showing you are 3 different synapses, 1, 2, 3, and 4. 1 and 2 happen to be excitatory synapses, 3 and 4 happen to be inhibitory. So if we were to activate synapse 1, we would see an EPSP, an increase in the membrane potential. If we activated synapse 3, which happens to be inhibitory, we would see hyperpolarization. If we activated the synapse number 1 twice, we would have temporal summation, and the threshold would be reached and an action potential would be generated. If we activated synapse 3 back to back, we would end up with a much greater hyperpolarization than if synapse 3 was activated only once. So here you're seeing temporal summation, but of an IPSP. Spatial summation in this case would be activating synapse 1 and 2, which are both excitatory. Separately, they don't do much, but when you combine their activity, you end up generating an EPSP large enough to generate an action potential. Similarly here, if we activated synapse 3 and synapse 4 separately, they generate, but they're inhibitory, they generate hyperpolarizations, but combined, the summation, the spatial summation of this hyperpolarization is much greater when the 2 synapses are activated at the same time. If we were to activate synapse 1, which is excitatory, at the same time as synapse 3, which is inhibitory, they would cancel one another out and you get absolutely no change in membrane potential in the postsynaptic cell. So hopefully this has kind of given you an idea of the ways in which combination of activity, again, onto a single neuron, can produce different effects or responses on the postsynaptic cell. So remember earlier I talked to you about axoaxonic synapses, and I'd mentioned to you the fact that at the level of the axon, when neurotransmitters are released, it's going to work to inhibit the action potential or even the release of neurotransmitters. And that's what we're going to talk about here. If we have a presynaptic cell releasing a neurotransmitter that produces a depolarization or EPSP in the postsynaptic cell, we would see something that looks like this in the postsynaptic cell. However, if we had a synapse right next to it that is inhibitory and generated a hyperpolarization or IPSP, collectively, right, the depolarization combined with the hyperpolarization would result in a flat line. If I were to draw it, would result in a flat line on the postsynaptic cell. This, when it happens, when an IPSP cancels out an EPSP, at the level of the dendrite, it's known as postsynaptic inhibition. However, you can have an axoaxonic synapse produce what is known as a presynaptic inhibition. In this case, the neurotransmitter being released onto the axon terminal here hyperpolarizes the membrane and prevents the neurotransmitter from being released at all. So this is known as presynaptic inhibition. So even before the neurotransmitter gets released, this presynaptic terminal ends up inhibiting or shutting down this presynaptic cell. In the previous example, the inhibition is happening at the level of the dendrite in the postsynaptic cell. However, in the second example here, it's happening presynaptically. So in this way, neurons can actually modulate each other and decide like, oh, you're activated too much, I'm going to shut you down, well before you even release neurotransmitters. So there's a check and balance happening in the nervous system that you might not have realized. So in this diagram, it's just showing you how the postsynaptic inhibition might work. You've got two different neurons synapsing onto the same cell. One's excitatory, the other is inhibitory, so you end up with postsynaptic inhibition. In this particular example, you have a terminal inhibiting another terminal. This is what we're calling presynaptic inhibition. We've kind of discussed the ways in which neurotransmitters produce different effects on the postsynaptic cell. We now need to talk about neurotransmitters and how they work at their receptors. So neurotransmitters, depending on what they are, can be packaged and synthesized very differently. Earlier we talked about two different synapses, those that resided in small vesicles and then those that had larger vesicles. They represent two different classes of neurotransmitters. You've got small and large. The small neurotransmitters have smaller synaptic vesicles, and they are considered to be classic neurotransmitters. These neurotransmitters that you've probably heard of, like serotonin and dopamine. Larger neurotransmitters are actually those smaller proteins that we discussed earlier. These are typically found in the dense core vesicles that I showed you in that photomicrograph earlier. They are made and stored very, very differently from each other. And we're going to talk about how they're different from each other in the next few slides. So first, let's talk about small neurotransmitters. So small neurotransmitters are those classic neurotransmitters. There's a ton of them, and in the second lecture for this week we'll be talking about some of those neurotransmitters. They're synthesized and packaged in the axon terminal or terminal button, so they're not actually made in the cell body, they're actually made on demand where they're released. The storage vesicles tend to be smaller because each molecule of small neurotransmitters tend to be smaller in size, so they don't take up a lot of space, and so their synaptic vesicles tend to be smaller in size. The vesicles are also located closer to the presynaptic membrane. They're the ones that get released first when an action potential reaches the axon terminal. Large neurotransmitters, because they are proteins, are actually made using DNA and mRNA that we talked about a couple of weeks ago. So large neurotransmitters are made in the cell body. They're all peptides. They're transported down the axon on cytoskeletal proteins. And then their vesicles tend to be larger in size, again, because proteins are just larger molecules in general compared to those classic neurotransmitters. The vesicles are located further away from the presynaptic membrane, so these neurotransmitters, or neuropeptides, are typically released when a terminal is really, really active. So the first line of release are actually small neurotransmitters, but if we end up activating that synapse again, it's likely that neuropeptides would be released. So peptides, just so you know, refer to proteins that are just very, very small. So they're like little baby proteins. They're called peptides. That's what I mean when I refer to peptides. And neurotransmitters that are peptides are known as neuropeptides. So let's talk about the way in which neurotransmitters are released, and this is true for both classic neurotransmitters as well as neuropeptides. The process of releasing neurotransmitters is known as exocytosis, and hopefully I'll drive this into your minds. Exocytosis is the release of neurotransmitters, and there are a few steps involved. The neurotransmitters are kind of, the classic ones, are located very close to the presynaptic membrane. We've got a huge difference in concentration of calcium, right? We've talked about sodium, potassium, and chloride so far, and we had mentioned calcium. This is where calcium fits in. It's a critical component of exocytosis. So calcium is present at a higher concentration outside the cell than inside the cell, and we've got voltage-gated calcium channels that are closed when a neuron is at rest. However, once the action potential comes down the axon to the terminal, it causes these voltage-gated calcium channels to open. Calcium then rushes into the cell, right, down its concentration gradient, and acts on the synaptic vesicles, causing them to fuse with the membrane and spill the contents of the vesicles into the synaptic cleft. So neurotransmitters are literally just getting spilled out into the synaptic cleft, and that's the process of exocytosis. One last piece, there's a calcium pump that takes the calcium that entered the cells and pumps it back outside so that it's ready for the next round of exocytosis. But that effectively are the steps involved in exocytosis. So again, just to kind of illustrate to you, right, I'm going to step back to the previous slide a second. When neurotransmitter vesicles fuse with the presynaptic membrane, they create what are known as omega bodies, these omega-shaped structures. And in this photomicrograph, you can actually see the omega bodies fused in with the presynaptic membrane. Isn't that incredible? So here you have, scientists have captured in real time the release of neurotransmitters, the process of exocytosis. This is by far my most favorite picture ever of exocytosis. What you're looking at here, right, is the bottom surface of a presynaptic terminal. So if you were standing on the postsynaptic membrane and looking up, this is what you would see here. You would see a whole bunch of dots, which are actually proteins embedded in the membrane of the presynaptic cell, and this space here where that looks pretty blank. This is the active zone. What you're looking at down here are the omega bodies. These holes that have been created are the holes of fusing of vesicles with the presynaptic membrane, right? Isn't that incredible that they captured it? So it's not visible right now, but these holes would be the spaces through which neurotransmitters would be spilling out into the synaptic cleft. So postsynaptic receptors. So neurotransmitters are not going to be able to send any message along to the postsynaptic cell unless the receptors for that particular neurotransmitter exists on the postsynaptic membrane. So each neurotransmitter actually has a collection or class of receptors, and each receptor within a class is known as a subtype. So you might have heard of the neurotransmitter dopamine. Dopamine has 5 subtypes of receptors, D1 through D5. Okay, so this is just an example. But receptors can be broken down into 2 functional classifications. They can either be ionotropic or metabotropic. Ionotropic receptors are known as ligand-gated ion channels. Right? Before this point, we've talked about voltage-gated ion channels that open when there's a change in membrane potential. Here, we have an ion channel that is gated in the presence or absence of neurotransmitters. The channel will be closed until the neurotransmitter binds to it. So these are ligand-gated ion channels, also known as ionotropic receptors. The other one is metabotropic receptor, and we'll talk more about these metabotropic receptors in a couple of slides. They're a bit more involved than ionotropic receptors. So the way in which ionotropic receptors work, or ligand-gated ion channels work, is that the neurotransmitter is released, binds to the receptor, which again, which is a pore in this case, right? Remember, channels are pores of proteins. But the pore is typically closed until the neurotransmitter binds, and once the neurotransmitter binds, it opens that pore. And depending on what kind of ion channel it is, if it allows sodium into the cell, if it's a sodium channel, it's going to produce an EPSP, or a depolarization, in the postsynaptic cell. However, if this ion channel happens to be a potassium channel, potassium is going to leave the cell, right, because there's more potassium inside the cell than out. Potassium will leave the cell, making the inside more negative, more hyperpolarized, or will generate an IPSP. So in this way, the kind of response in the postsynaptic cell depends not so much on the neurotransmitter actually, but on the receptor to which it activates. If that receptor is a sodium channel, it's going to generate an EPSP. But if that receptor happens to be a potassium channel, it's going to generate an IPSP. So most people talk about neurotransmitters as producing effects, but really the thing that holds the key to how or the kind of response a neurotransmitter will have depends on the protein, the receptor in the postsynaptic cell. So this is an image from your textbook. Ligand-gated ion channels bind ligands or neurotransmitters, and they open the channel and allow ions to go through. In the absence of neurotransmitters, the channel is closed. So again, just to illustrate how sodium might produce an EPSP, if you've got neurotransmitters binding to the receptor and opening these channels, sodium enters the cell and makes the membrane more positive to generate an EPSP. If you've got a chloride channel, neurotransmitter binds to this receptor, chloride enters the cell, right? Chloride's a negative ion, so it's going to make the membrane more negative and generate an IPSP. So again, the kind of receptor on the postsynaptic cell is going to determine the response generated in the postsynaptic cell. The second kind of receptors that we're going to talk about that are a bit more involved are G protein coupled receptors, or metabotropic receptors. These receptors are proteins, but they're not a channel or a pore. They're just a hunk of protein hanging out in the membrane. So in this case, the neurotransmitter binds to the receptor, this G protein coupled receptor, this hunk of protein, but this protein happens to have a specialized protein attached to it called the G protein. Now you know why it's called a G protein linked receptor. These are also known as metabotropic receptors. So when the neurotransmitter binds to the receptor, it activates this G protein. And what this G protein does is actually quite diverse. It can do a whole host of different things. It might activate an ion channel and allow sodium in, causing the cell to be depolarized. It can generate what's called a second messenger. So the neurotransmitter is the first messenger. It can generate a molecule called a second messenger that carries the information further into the cell. And this second messenger can change the expression of genes, it can change the metabolic activity of a cell, or it might even open or close an ion channel. So you can see the diversity of response is much greater for metabotropic receptors than it is for ionotropic receptors. Ionotropic receptors are kind of just like a one-trick pony. They open when the neurotransmitter binds, and they shut when the neurotransmitter leaves. There's much more opportunity or options that can exist in the postsynaptic cell if we're talking about a metabotropic receptor. The only difference is that ionotropic receptors produce an immediate response. It's a very, very quick response, but metabotropic receptors, because there are so many steps involved, the response might be very delayed. But if you're going to change things like metabolic activity or gene expression, the effects produced by a metabotropic receptor are going to be much, much longer lasting than those that you might find with an ionotropic receptor. And again, this is just another example of how metabotropic receptor works, or G protein coupled receptor, an image from your textbook, but it's effectively saying the same thing. The receptor has a G protein attached to it. This G protein can produce second messengers that can affect the activity of ion channels, but it might also change the expression of genes as well as metabolic activity. So in your textbook, this is an image that kind of puts everything that we've talked about so far in this lecture. The synaptic vesicles are kind of hanging out in the terminal. Calcium enters the cell and causes the vesicles to fuse with the membrane. Ultimately, the neurotransmitters spill out. They bind to receptors in the postsynaptic cell, allowing for ions to either move into or out of the cell. The influx or entry of ions or exit of ions generates either depolarizations or EPSPs or hyperpolarizations or IPSPs in the postsynaptic cell. One last piece of the kind of story that we need to complete, and that has to do with the removal of the neurotransmitters. So neurotransmitters can't keep hanging out and binding to receptors on the postsynaptic cell. They have to do their job, and then they come off of it, come off the receptors, and then move on to their merry way. The nervous system is very efficient. It does not waste. So what happens to neurotransmitters is that they typically get recycled in some way. So here's our presynaptic terminal, our postsynaptic cell. The neurotransmitter gets released, activates the receptor, and then comes off the receptor. So it can be broken down in one of two ways. One is that an enzyme that hangs out in the synaptic cleft ends up taking this neurotransmitter and breaking it down into smaller components that are not active, and then these components are either recycled back into the terminal and reused to make new neurotransmitter and repackaged and so on and so forth. The other thing that can happen is the neurotransmitter can be pumped in whole or as a whole into the presynaptic terminal through a protein known as a reuptake transporter. So one mechanism is enzyme degradation, and the other one is the reuptake mechanism. Either way, the neurotransmitter ends up getting repackaged into vesicles and then ready for re-release. Okay, so this protein is known as a reuptake transporter. So we're going to spend the next couple of slides putting everything we've talked about over the last couple of weeks together to make sense. So we've kind of talked about action potentials and the membrane potential and neurotransmitter release and receptors. And I want you to recognize that all this information that we've kind of been building over the last couple of weeks works together to allow the nervous system to function the way it does. And so I wanted to put it all together so you can see kind of the big picture of why we went over all that we did. We're only going to be talking about sodium as an ion because it's so much easier to kind of have a one option rather than consider all the many different ways that a neuron can respond. So we're only going to be looking at EPSPs and the influx or the entry of sodium into the cell. All right, so you know that an EPSP is a change in membrane potential towards a positive value caused by sodium entering the cell through ligand-gated sodium channels. The sodium ions are then going to diffuse down the dendrite towards the soma, leading to the passive movement of charges, right? So they don't get propagated, but they passively diffuse to where there's less of them. These positive charges from multiple different synapses can be combined into one overall positive charge through the process of summation. If this larger combined positive charge reaches the threshold of excitation at the axon hillock, you're going to have voltage-gated sodium channels opening. At this point, tons of sodium enters the cell, making the inside of the axon much more positive and begins the action potential. So the action potential moves down the axon through the purposeful opening of voltage-gated sodium channels, right? This is a more active way. The only way the action potential propagates down the axon is if voltage-gated sodium channels keep opening down the length of the axon. Once it reaches the terminal, once the action potential reaches the terminal, it's going to cause voltage-gated calcium channels to open. Calcium enters the axon terminal because there's much more of it outside the cell than inside the cell. This calcium influx causes the vesicles containing neurotransmitters to fuse with the presynaptic membrane, releasing neurotransmitters onto the synaptic cleft. These neurotransmitters bind to ionotropic receptors, which are ligand-gated sodium channels, letting sodium into the postsynaptic cell, which causes an EPSP. And so hopefully you can see that we're kind of back to square one. That's where we started, but we kind of put it all together in allowing you to hopefully see how neurons communicate. And so this is kind of an image, my sad animated attempt at getting you to see the bigger picture. What we have here is what's happening at the level of the dendrites. So this is the neurotransmitter, and these are sodium ions. The outside's more positive relative to the inside. So the neurotransmitter binds to the ionotropic receptor, which causes the channel to open. Sodium rushes into the cell, producing an EPSP at the level of the dendrites. LG stands for ligand-gated. VG stands for voltage-gated. So only voltage-gated channels can be found at the axon, and typically only ligand-gated ion channels can be found in the dendrites, soma, and axon terminal. So these ligand-gated ion channels that are happening all over the dendrites allow sodium into the cell, right? Sodium from all these different areas combine together to a much larger charge, positive charge, through summation. Hopefully you recognize that this was spatial summation. At the axon hillock, if this overall charge reaches the threshold of excitation, an action potential will be generated. The action potential will cause voltage-gated sodium channels to open, propagating the action potential down the length of the axon to the terminal, where voltage-gated calcium channels will open, causing the fusion of synaptic vesicles with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. Okay, and starting the process all over again, but just for this particular neuron. Okay, that's it for this lecture. Don't forget to submit the reflection. I know that it had a lot more material than in previous lectures, but it's kind of really hard to talk about all of them in isolation, and I wanted you to see how it all fit together at the end, what we've been doing for multiple weeks. So again, don't forget to submit your reflection online.